point where an investor shows up on the curve. And I think what she left out was in between there's a customer somewhere, okay? So we have to find the customer, and you have to understand where they are hanging out, what they're thinking about. So here you have laboratory flow, sample prep, diagnostic assay, clinical evaluation, and then you have a clinical diagnostic flow. They use the laboratory service, but I don't think they really care really what they're doing. They want information. And you start to throw things in the space there of how people are behaving, and then again, some more gaps start to pop out, or ideas, and you start to say, well, do those make sense? What would we do with them? Maybe there's a good reason that gap's there, that there's no value there. So we spend some time to look at that. Then maybe as you get along with that and you do enough reading and you're talking to people, you can start to generate that there's a list of needs that have to be addressed. So you've identified, you've segmented, you've kind of targeted people you want to talk to, and you can start to say, well, what are their concerns? And if you don't go broad enough, you may fall in love with a certain population and a certain list of concerns, but it's not adequate. And so you have to go much broader and to map that side of the problem. So we spend some time on that. And then you start to say, okay, now we come back to systems, and this stuff starts to reintegrate maybe. So here I have somewhat clip art images, but start to pull together that, well, how does this flow? And let's revisit how the technology would fit in a flow. And you come across two important things to think about. One is clinical utility, and the other is medical utility. And clinical, medical utility, focusing on that is really what is the patient going to get out of this? And how is their life going to benefit? And then in this image, we look again at how that maps into what businesses are doing and how the community is trying to perform. And what you see is that in this bottom flow here, I think we have really some great work being done on advancing morphometric analysis. But from my view of it, from kind of a process view, it has high value in operational efficiencies and operational improvements, and those are have the companies that have large market share, that are well entrenched. The laboratories using their technology will value that. However, on the operational side, you get into operational improvement problems because you want to save money. So you don't necessarily want to pay a lot of money for the technology to save money. And so I think there's going to be a battle there that you're going to face. And if that's the battle you fight, there is opportunity. Our organization doesn't see our contribution being strong in that type of battle. That we really want to be on the more front end of the curve, in the darker part of the valley, hopefully seeing a path to come out, and with a willingness to take some risk to travel that path. And then the last thing I'll come back to, because I think it's very important, is this is very busy, but if you look at the World Health Organization, their small cell lung cancer protocol was revised again in 2010. If you go and look through that in the pathology section of that, the standard is still something published around 2002 or 2003 on the categories of typing of the tissue. So you kind of have to understand, at least in this problem, I think, is one answer enough? And I think in this picture here, the answer may be no. There's at least 8 major categories and multiple subcategories. So another vetting exercise has to take place to say, well, is there an 80-20 rule in here that the industry would value that would add clinical and medical utility? that has to be sorted out. But then you say, well, why would I go through all that effort? And you check yourself another way, and worldwide you see that early diagnosis is definitely a place that people are interested in. That some of the publications say 90% of the cancer can be halted in its course if picked up early enough. And parts of that are technology, and there's a lot of great technology we saw today where people are trying to get at that, and part of it is knowing your population, and can you segment and identify the population to apply that technology. And much of the literature around the DNA, microarrays, gene expression arrays, and that, if you really dig into that, you're going to see that part of why they may be stalled to the point of really broad and success. And someone said, I think earlier, it was only 20 biomarkers that have really grounded, is because they are struggling to segment the population, identify a target group that they can tie the marker to a therapeutic action, to medical utility. I don't know why they haven't done more in that area, but the literature is peppered with criticism and concern that too little has been done. And then the last thing, in looking at the problem in the cancer space too, is that localized, regional, metastatic, these are another dimension on the problem. And what part of that are you looking at? How is the problem defined? How does the clinical community recognize that problem? And can you segment within that? Can you pick a couple sections or spots on that matrix? And is that a business? Can the technology be tested and proved there, or do you have to do the whole matrix? And again, these are problems that we look at in making our decisions on what we want to work on. And I think what we've come to a conclusion is that there's a lot of hard work ahead, but the cancer diagnostic space has some big gaps, and these gaps are definable, and they can be addressed with a rigorous plan and the right technology put in place. And in our review and discussions and work with Max's lab, we felt that he was really heading in the right direction on the technology side. So we hope to bring the rigor on the other side from our company and what we do.